But without further ado, Dr. Gregg Dean is here. You guys may remember him as the head of our Small Animal Advisory Board. He's just retired, but we wouldn't let him go away. We made him come in and talk about feline coronaviruses, which you guys know, FIP. Gregg's he had to spell this out for me. He's the professor and the head of the Department of Microbiology, Immunology, and Pathology. I saw MIP. I started to think of all these little acronyms like Men in Pathology, or Men in Purple, or whatever. So Gregg's going to talk to us, and as many of you guys know, FIP, which we funded a lot of work on FIP and feline coronaviruses. So Gregg was gracious enough to come and talk to us. So take it away, Gregg.
All right.
Thanks,
Thank you. Thank you for the invitation, really. It's always a pleasure to come down and see all of my friends here at MAF and talk about something that I love to hate, I guess. It's such an interesting and tragic disease in cats. And you go through a career, you try to think about what your impact is going to be. You hope you're doing things on a daily basis that are impactful, but I do research as well, and I've always felt if I could pick one thing to choose and feel like I was very successful through my career, it would be making a positive contribution to the diagnosis prevention of FIP. It's a very interesting virus. So I'm in the Department of Microbiology. That's where I got my microbiology degree. I also did a residency there too, so I'm a pathologist. So from the scientific point of view, it's really interesting. The virus is very interesting, but the clinical disease is very devastating. And I really view it as truly a Jekyll and Hyde situation, and I'll try to explain a little bit about that. Just stop me if you have any questions. I'm rather have it more conversational than just me talking. So these are my 5 cats. I actually put together this presentation shortly after MAF funded this project I'm going to talk just a tiny bit about today. Funded it about 2 years ago. And the student organization, veterinary student organization, asked me to talk about Morris Animal Foundation and about this research. And we were just getting ready to launch it, so it was exciting. And I put this slide together. So these are my 5 cats. I need to revise the slide because there's been some changes. And I have to give a little biography on each of them. This is Mr. Magoo. He is the most beautiful Siamese cat. He was an owner-relinquished animal. He was about 18 or 19 years old. It was a pretty tragic situation. The woman who had owned him his whole life was not doing well and had to go into a nursing care facility. And so she relinquished this cat. And it's not easy to place a 19-year-old cat. So he joined our house. He lived 5 more years. We never thought he'd go that long because when we first got him, we knew he was hyperthyroid and he had renal failure to boot. But he lived 5 more years. My wife's also a veterinarian, and she is really good. And she can extend any animal's life. But Mr. Magoo passed away from his chronic renal failure a few months ago. So he's the only male. The rest are all the girls. This is Ed. Yeah, she is a girl, and she embraces her name fully. She's had an interesting life, really struggled with atopic dermatitis. So she's an itchy cat. And she would just pull her fur out. She was a mess, really. We treated her very successfully, though, with cyclosporin for the most part. And you can see, she had great body condition and a really nice coat. It just was truly a lifesaver for her and gave her real quality of life. And just in the last month, she came down with renal lymphoma. And so she's no longer with us either. So we're really down to these 3 girls. This is Elsie, named after my grandmother. I found her running in a state park in North Carolina. She was just a tiny kitten loaded with fleas. And I don't know how she got out there, but it was no place for a kitten. With the predators, it's bizarre that she made it as long as she did. But I took her, and she has grown to quite a sizable cat. She's a little difficult to manage that way. This is Phoebe. She is just a happy-go-lucky cat. She's writing a novel. Places I love to sleep. And she spends every day exploring new and interesting places and evaluating them for all the key criteria that a cat should take into consideration. And then there's Izzy. Izzy, who came out of the Adams County shelter, and she had been adopted and returned 3 times. She has a bad habit of biting any hand that is put in front of her face. So not the pet that apparently at least 3 adoptive homes thought they wanted. Again, my wife, she was a basket case, pretty nervous and wasn't eating, and quite anorectic, and they were ready to euthanize her at Adams County. And my wife couldn't allow that to happen. So she joined our house. She has quite a personality. You don't just put your hand down in front of her face. But she will invite you to pet her. But it's by invitation only. She really is a nice cat, though. Okay, so I wanted to just talk a little bit about coronaviruses because they're super interesting, like I said. They're very pretty. So this is an electron micrograph, a color-enhanced electron micrograph of a coronavirus. So they get their name from these spikes that are on the surface, quite dramatic, actually. And I wanted to present this just so you can make the connections that these are viruses that are found in so many different species and cause significant disease, especially when they jump suddenly into a new species. And so some examples in humans are the acute respiratory syndrome that cropped up a few years ago, severe acute respiratory syndrome, and then the Middle Eastern respiratory syndrome. So 2 severe respiratory diseases, new to us, caused a lot of trouble, shut down a lot of airports. So humans, poultry, cattle, pigs, dogs, mice have their own coronaviruses. And typically when you look at coronaviruses, they cause disease in the respiratory tract and in the GI tract. That's most common. Bats seem to carry these viruses. They're good at being a reservoir. Apparently it's asymptomatic. I would say, though, that we don't know that for sure. Cats, though, is a really different and interesting story, that we have this Jekyll and Hyde situation of a fairly simple enteritis and then this very fatal infectious peritonitis. So this is a bat. Such interesting creatures. So common features that you see with the coronaviruses is that they circulate in populations. So they're always there. There's always somebody infected, somebody shedding the virus, new people getting infected. For the most part, everybody exists happily together. But it persists in the environment and it's highly contagious. So it's actually a pretty successful pathogen. It replicates well, it's transmitted easily, it circulates in the population, usually without killing individuals, and so it's a mutually beneficial interaction. So usually mild symptomatic or asymptomatic symptoms. There is this zoonotic potential, though, and that's when this virus can jump out of one species into another, into humans, and then we see SARS and MERS, and it's a big deal. There are successful vaccines in different species, so it's not as if you can't vaccinate successfully against a coronavirus. So just to focus on feline coronaviruses in particular, pretty interesting. It's estimated to have entered the cat population about 100 years ago. And there are these two biotypes, so two disease manifestations that we see, the enteric form here and then the infectious peritonitis here. And I have to put a picture of Nels Peterson up because I did my postdoctoral fellowship with Nels Peterson, and he's trained a lot of scientists over the years, and I'm sure many of you have probably met him before, know of his name. He is a big guy. He's Danish. You put a kitten in his hands and he turns to mush. It's pretty funny because he's kind of a big, tough, gruff guy, but yeah, he melts when he has a kitten. And he has been a real driver, I would say, in the study of feline coronaviruses over the years. Anybody who passes through his lab gains some appreciation and desire to do something about it. So I owe Nels a lot, but also owe him my interest in doing something about feline infectious peritonitis. So Dr. Jekyll, that's the enteric form of this infection. Again, it's found in cats around the world, except interestingly, and these are always the facts you can share at cocktail parties, maybe Thursday night trivia at the local brewery. I don't know if this one will ever come up. Not found in the Falkland Islands. Pretty bizarre, actually. Typically asymptomatic. So here's a kitten with enteric coronavirus infection, shedding the virus. You can't tell one way or another. Maybe you have loose stools or diarrhea, but it's usually asymptomatic. But the virus replicates, reproduces itself in large number, and the cat shed it. And so then it's transmitted very easily, fecal, oral, through the saliva. So again, very, very successful microbe. Mr. Hyde, feline infectious peritonitis virus. And this is really fascinating and what distinguishes the feline coronavirus from other coronaviruses of other species. And FIPV arises from mutations in the enteric form. So cats get infected, virus replicates, sometimes it mutates, and it results in a virus with different characteristics. And causes severe disease that can be categorized by these forms, and it's really associated with both the clinical presentation and then the targeting of the virus. So there's a dry and a wet form, and I'll talk a little bit more about that. There's also a neurologic form, and then there's uveitis, so inflammation in the eyes. Again, it's usually fatal, and there's two things that I've always two diseases that have been most impactful when I see animals that have it. One is heat stroke in dogs. That's a horrible disease or situation. And then the second is feline infectious peritonitis. I don't know, there's something about those two things that evoke the most emotion in me. Usually fatal, very difficult to diagnose. And this is a frustration for sure, and I'll come back to that. And there's no uniformly effective treatment right now. This is a Siamese cat, and it's hard to appreciate, but the abdomen of this cat is quite round, and that's because of the fluid that's exuded into the body cavity. So,
are you saying that that normally it's from spontaneous mutations?
Yes, in individual animals.
So does it evolve rapidly in general, the coronavirus evolve rapidly?
They do. So it's an RNA virus, so its replication is error prone. So you see a lot of mutations. So you can look at almost any cat and you can find mutations, but it's having the mutation that allows the virus to expand its scope of infection, and that the cat is unable to defend against. Yeah, so you have to have the combination of those things. Yeah.
When it's mutated like that, is it as transmissible as it was before, like cold?
Yeah, it can be transmitted. And you'll see these micro outbreaks of FIP. And there's debate that goes on about what's transmitted and what is a spontaneous individual mutation in a cat.
So it's unclear how much of it jumps from cat to cat.
Yeah, and when you see these outbreaks in a cattery, a closed population, it's hard to wrap your head around the fact that you've had multiple simultaneous mutations in a few cats. That's probably transmission from cat to cat. Yeah.
Like they might have all been infected with a version of the virus that's just about ready to mutate if one little thing goes wrong, say, and then it does in 4 cats.
And it does what?
And then it does in like maybe several cats.
Yeah, interesting idea. So you've got an evolution within a closed population that's bringing it close to that place where a particular mutation will allow it. It would be super interesting to study these things. You can imagine it's difficult because you can never predict prospectively which animals are evolving this virus. There's some studies going on now trying to look at that a little bit closer. Hard to follow the right animals and catch them at that moment of mutation. Any other questions? Yeah, so the mutation allows the virus to infect a different category of cells, cells that circulate around the body. So instead of being contained within the GI tract, now the virus is distributed to more tissues. And you get this systemic distribution. And it causes inflammation of the vasculature and allows fluid to come out of the blood vessels, and that's how you get this effusion. And it's associated with this granulomatous inflammation, and I'll just point some things out here. This is a cat's body cavity, their intestines, and you can see this fluid, which isn't normal. It's classic, it's a classic appearance for FIP. And the same thing over here. And these are inflammatory cells actually in the central nervous system. Not normal, that's how you get the neurologic form. And you can see these little white nodules on the surface of the intestine. Those are little foci of inflammation, granulomatous inflammation. And then this uveitis is also pretty commonly associated with FIP in cats. So that's what it looks like, pretty devastating, affects multiple organs. And the idea, so the question is why? Why can't the cat manage this? Cats get infected all the time with enteric coronavirus. Why can't they manage this one? What is the difference in an immune response that's allowing this to happen? And a lot of work over a number of years has really identified a particular aspect of the immune response. So there's a cellular response where an immune cell can kill a virus-infected cell, for example. And then there's the humoral side, so that's the antibody component. So antibodies are produced, it recognizes the virus, and that can also be an effective way to control a pathogen. In this case, it's thought that that cellular response is what's lacking. So the disease is just so frustrating from every perspective, honestly. Diagnosis and treatment, really challenging. So from the diagnostic point of view, most of the tests are indirect. There's not really an easy single test that can be reliably used in all cases to make the diagnosis. So sometimes the symptoms are vague and can carry on for some period of time, and you just can't get to the diagnosis. Hard on the cat, of course, difficult for the owners, frustrating for the veterinarian. So most tests are indirect, but there are PCR-based tests. Those are helpful in some ways, can be confusing in some ways.
Sorry, you define PCR.
Yeah, sorry, polymerase chain reaction. So this is a way to amplify the viral nucleic acid. So it's a very powerful technique. It's very sensitive. It tells you, yes, this virus is there. Okay, so you're actually looking for the genetic material of the virus itself in an animal.
Thank you. Are there times when that type of test wouldn't work?
Sure, you've got to have the right cells to go in the assay, so that's a problem. Sensitivity could be a problem, but then we've also got this huge problem that most cats have been or are infected with the enteric coronavirus. So how do you tell that enteric coronavirus from the one that causes FIP? That's very challenging. And there's been some advances there, but again, we don't have a uniformly successful diagnostic test.
Is this one of those where you know that there's like these few genes that you associate with it, but they just might not always be in the same place at the same hours?
The mutation?
Yeah.
Yeah, and that's, again, another pretty intense area of focus. Can we identify the few mutations associated with the change in disease type, the biotype? And there's been 3 that are pretty clearly associated. Are they the only ones? That doesn't seem to be true. So, and then you get into the problem of what if it requires multiple mutations? So that's always hard to get to know that, okay, this pair or this pair can give you the same end result.
But the upshot is even if you get the virus and you can multiply it, it's hard to tell the difference between it and another virus.
Can be. And sometimes it's just by where it is. So like I said, the enteric coronavirus is usually in the intestine. FIP is circulating. But what we've learned by going down this road is that you can find enteric coronaviruses in a lot of places too. So, because the technique is so sensitive. So, very challenging. The other approach is to actually look for viral proteins. So have some of that inflammatory tissue. There's definitely virus in there. So if you get some of that and you stain it, you can identify virus. I'll show you a little bit of that. The treatment has always been just symptomatic, and this is how it starts out. It's a sick cat with one thing or another, and fever, and so maybe dehydrated, not eating, all of these issues, maybe uveitis. So the treatment often starts out as symptomatic, just trying to deal with what's causing the discomfort in the cat at the moment that it's presented, but really there's no uniform treatment that has been identified yet to actually reverse the disease. There is some really exciting work that Nils Peterson is involved in with an antiviral small molecule. So it's super exciting, and they're working to develop analogs of those antivirals that hopefully will be more uniformly efficacious.
Does it matter the age of the cat? Do younger cats turn to FIP a lot more than
Yeah, they do.
older cats?
So typically under 3 years of age. Yeah.
So like a 9-year-old cat generally wouldn't.
Generally wouldn't, but I guess when
Not impossible.
not impossible for sure. Yeah.
And when it's misdiagnosed, what are the common
Their
Diagnosis.
Yeah, so you probably have a boatload of these.
When I would see these, is it just a run-of-the-mill diarrhea? Is it, I hate to say it, but lymphoma?
Yeah.
And sometimes I would hope it was lymphoma
Mm-hmm.
versus FIP, because lymphoma I could do something about
Yeah.
for a while. I mean, that's how crappy this disease is when you wish cancer on a cat versus FIP. And, I don't know, trying to think of other viral diseases. Usually I would think is it something GI? Is it something the cat got into? But that's, the wet form is one thing. The other is the dry is way harder
Yeah.
to figure out. And that's where you're looking at lymphoma and fungal diseases if you were in certain areas of
Right.
the country. So nothing good. Very few things that are good. Did I cover probably?
No, you did. The neurologic forms can be frustrating. The symptoms can be subtle and hard to pin down based on a neuro exam. And so you go through the same thing. Maybe it's toxoplasma or some other infectious agent or cancer. Liz, you got any to add? No?
And the uveitis, you might think it's just a herpes virus
Yep.
or something really simple to begin with, and so they can look like okay, that's what's going on, and then they don't, it gets worse.
Yeah, it just keeps going and you can't seem to get on top of it. And that's when, as a clinician, you start, your suspicion starts to go up. And again, with the dry form, very difficult. Weight loss, and so you pursue all those issues related to weight loss. I mean, you just are literally chasing your tail trying to figure out what it is in some cases. When they present and their belly is full of fluid, like I showed there, that's usually pretty easy.
I've explored some of these cats to try to get a diagnosis, which is pretty extreme.
Yeah.
To try to get a piece of tissue to look at, and then it's more obvious, but it's still, that's invasive diagnostic test. Right.
So I wanted to just take a second because Kelly Santangelo and I are funded by Morris Animal to look at some diagnostic strategies here. And Kelly is really focusing on immunocytochemistry. So the advantage here is that we already get these samples oftentimes as part of the diagnostic workup, and so if we could make use of those instead of having to go to a exploratory surgical procedure or some other invasive biopsy strategy, if we can just get some of these cells and get them on a slide, maybe we can identify virus inside of them. And so we're thinking about is particularly cells out of the abdomen or out of the chest. And that's what's shown here. And so this is pretty, so it's nice. These are nuclei of cells that are stained blue, so you can see where the cells are. These cells are stained for the viral protein, so they show up green here. And then this stain tells us the type of cells that these are. They're monocytes, and then we can overlay all of those and show that we've got virus within the monocytes. And that's very helpful diagnostically in some cases. The other thing that we're doing in this study is really a discovery project where we're looking at plasma biomarkers, and there's over 1,300 biomarkers that we've now run. I just got the huge spreadsheet back with all the data. So we're basically hoping to identify a few changes that we can see in the plasma of infected cats that tip us off that it's FIP, something that's unique to FIP. We haven't begun to crunch the data on that one, so we'll see where it leads us.
So I have a quick question. Is this from cells from a fusion?
Yes.
Okay.
Yep, this is an effusion. Yeah, so they really light up. This is a dramatic case.
Do you use one stain, take a shot, stain the same sample again, take a shot? Or
All of the stains go on at the same time. It's just filters. So we've got a filter that looks at the green, one for the red, and one for the blue, and it overlays. Yeah, so the staining all happens simultaneously.
So it's one of the things you're looking for, the idea that there's something different about certain cats that cause them to be able to get this thing?
Right, so there's been a lot of work. I think Nels Peterson and Leslie Lyons have worked together for quite a while looking at the genetic basis, because you do see a disproportionate number of particularly purebred Persian cats, for instance, that tend to show up with FIP. And so, you know, I don't think they have any doubt, and I don't really have any doubt that there is a genetic predisposition that probably explains the inability of the immune system to respond in an effective way to prevent the spread of the virus.
And do we know whether it's Persian cats that are more likely to get it or expensive cats?
Well,
I mean,
yeah, no, it's fair
spend
enough.
A lot of money on a purebred cat might be more likely to take them to the vet and go through a lot of
Oh, yeah. So is it just that we're not making the diagnosis on your farm cat? And yeah, I don't think that that's it. So, but you do see a higher likelihood in some of these catteries. Missy, do you have a comment? Yeah. Oh.
Not just.
Yeah, Missy used to work at the Humane Society, so you can comment. You saw a lot of kids. So.
Yeah, a lot of kittens with FIP.
Yeah.
And it wasn't just purebreds, it didn't seem like to me, but we didn't have purebred cats. Right?
Yeah. Right.
Yeah, I was just curious whether it's something that might be more around that people don't diagnose or something.
Yeah, no, I think we see it in all sorts of cats. So vaccines, this has been a real frustration. Obviously, we'd love to have a vaccine to prevent this. And there have been many attempts, actually, and even some products on the market. They're not very effective, and they're generally not recommended. And the problem here is actually also very interesting. So as I mentioned before, there are vaccines for other species, dogs and humans and pigs. Those tend to be vaccines that are prepared either from a modified live virus or the virus is just killed and injected. So pretty standard, often very effective way to generate a vaccine, but it doesn't work here. And the reason that it seems to fail is because the cat's immune response against that vaccine actually then predisposes them to infection. So that's not at all what you want to achieve with a vaccine. And what this little cartoon shows here is a cell, a monocyte for instance, and virus here, and then an antibody. So if you immunize a cat, they generate this antibody, they're exposed to the virus, and the antibody actually ushers that viral particle to the very cell that it wants to get into so that it can go systemically and cause disease. And so that's where you get this idea of enhancement. It's not the only place. Cats are very interesting because they have a real tendency to do this, more so than other species. We've seen it with FIV as well. But it's also very similar to what happens with dengue. So dengue is a very common viral disease in humans, and there's 4 serovars. And if you're exposed to one and generate antibodies, there's a good chance that those antibodies will enhance your infection if you're infected with another serovar. So you get this enhancement phenomenon, and actually dengue can look a lot like FIP. So it's effusive, it causes a vasculitis and fluid losing disease. So this is what we have to avoid. And you know, our goal, you know, because I've struggled with this for a long time. What do we do? What do we do if we can't find a way to vaccinate against this? And you know, I really started to think we're targeting the wrong time point. We have been targeting Mr. Hyde over here. So waiting until this emerges and then trying to treat for it, or vaccinating against this and hoping that it actually works, which we know it doesn't. But what if we go after Dr. Jekyll instead and head this off early? So to do that, you have to understand how this virus exists in a cat population. And some of this, I mentioned before, the virus is circulating in cat populations all the time. And the reason it can do this is because we have all these different states of the cats, some that have never been infected, some that have active infection. So the virus, they've got the virus, it's replicating. You can see that they're making an immune response. You've got some that are convalescent. So they've got antibody, but no virus. So presumably, they've gotten rid of the virus with their immune response. But we also see chronic or intermittent shedders. So cats that just episodically shed the virus, or that just shed it all the time. Whenever you test them, they're shedding virus. So it's sort of the typhoid Mary, because they're perfectly happy, but these are the ones that are in a population that the virus is using to make sure there's always a source of virus for any susceptible animal, which might be this uninfected one up here that's never seen it, or it could be one that is then susceptible to reinfection. So the approach that we decided to use, and this is the hypothesis that we actually put in the grant, I rephrased it so it's a little more straightforward. So, you know, really, if we can understand how the immune system in the intestine is controlling that enteric coronavirus, maybe we can develop a vaccine to eliminate the Dr. Jekyll and avoid the Mr. Hyde. Easier said than done, but the study has, now we're completely done with this phase of it, and it's been fantastic. And I just want to point out the impact, what it takes to do a study like this, give credit where credit is due, and then introduce some of the players. So this is Morgan Pearson. She was a veterinary summer student. She came from Iowa State to work in my lab for the summer, and she jumped right in with this biopsy tool and did the biopsies on all these cats. So we took colonic biopsies so we could get a sample of this tissue and understand what's happening in the colon. We had all of the internal medicine folks over there, every one of them at one time or another came to help us. They were phenomenal. Multiple undergraduate students, but this student in particular, Mackenzie Smith, she was fantastic and ran a bunch of assays for us. Really very nice to work with, and now she's in school to be a physician assistant. So, and then our laboratory animal interns, they were very interested in this project and helped us out. I have a graduate student, Allison Vielander. She's an anatomic pathologist. She's helped us. And we've got a clinical pathology resident, Samantha Evans, who has also helped us. And Samantha is going to present all of the data that we have generated so far at the International Society for Companion Animal Infectious Disease in a couple weeks. Yeah. That's up in Portland this year, and so Sam is going to present everything there. But the way we wanted to tackle this was to actually look at that immune system in the intestine. The immune system sort of split into two parts: the mucosal immune system, so in the GI tract, in the lungs, in your conjunctiva, urogenital tract. And then the systemic, so you feel your lymph nodes, spleen, everything in your blood. Those systems certainly talk to each other, but they are fairly separate as well. And just by looking at one, you can't necessarily understand what's happening with the other. So our goal with this study was to utilize a colony of cats that we have at CSU, where we have circulating enteric coronavirus, and to collect colonic biopsies, and understand that immune response and how it's controlling the virus.
Is that Dr. Webb?
Yes, Craig's fantastic. And just as a sidebar, Morgan loved this project so much, she's taken a break from veterinary school and she's doing a master's in the lab because she wanted to see this project to the end. So it's pretty cool. She's going to be a star. And Craig is already a star. So this is data, and I don't want to numb your minds too much with it, but I just put it up here. It's just a small bit of what we've learned, but really key. So before I told you that what was suspected as far as the immune system not controlling the systemic infection of FIPV, what we found with the enteric coronavirus, and this isn't terribly shocking, but it is the antibody that's found in the GI tract that's controlling the enteric coronavirus. The cell-mediated part does not play a role here. So, and these are just two different types of assays that look at immunoglobulin A, which is the most common type of antibody in the GI tract. And this is our group that is both, that is replicating virus. So these cats are not infected. These cats have been infected but do not have virus. And then these cats have virus and are responding to that infection. We really got lucky on this study. We had 9 cats that we caught right at the point where they flipped from replicating virus to no longer replicating virus. Just dumb luck. We looked at 34 animals total. It really gave us a unique opportunity to catch them at that key point in time. And this is kind of fun because I'm a scientist too. I love, this is how we find out what these cells are doing. So we can put cells on these membranes, they make this antibody, and we can stain the antibody. And so you can actually count the cells that are producing antibody against the virus. So it's kind of fun. So we're very excited about this because now we have some idea of what it would take to immunize cats against the enteric coronavirus. We're ready to take the next step. And just super briefly, we've also been working for some time to use Lactobacillus acidophilus. So this is a probiotic bacteria that's in your yogurt and lots of other things to deliver a vaccine orally. So instead of injecting a vaccine where you have a chance at, injection site sarcoma, really we want to deliver this orally. We want to get right at inducing the immune response where the virus is going to come in. And so and we hope to hit this balance of a really safe vaccine, very effective vaccine, so that if you think about that shelter environment that Missy's worked in and cats constantly streaming through, stressed, it induces shedding of this virus, new cats are getting infected, some of them go on to end up with FIP. If you could immunize them very easily when they come in the door with an oral vaccine and hopefully suppress, help them control the enteric coronavirus very quickly, then hopefully you don't get that experiment that the virus is doing and producing a mutation that results in FIP. So that's the hope. But really, I have to thank all you because I'm so appreciative of all the work you do every day because you care about animals, you do what it takes to go out and find those donors who are willing to support this type of work, and hopefully on our end we can do the best possible job with the money that we were granted and make a difference. So thank you.